My original interest in psychic A&P came from following the work of Earl Sutherland, who was studying the mechanisms by which the hormones glucagon and epinephrine cause the breakdown of glycogen to glucose in liver cells. And he came out of the Corey school and was interested in the mechanisms of hormone action, and I had no particular interest in that subject in relationship to my own personal work, but it just interested me from an intellectual point of view. And so I followed his work pretty carefully. This is while I was a graduate student in the early 50s at Johns Hopkins. And I continued to follow his work during the 5 years I was in England doing postdoctoral studies, and then in the 8 years or so that I was at the Geigy Pharmaceutical Company after my return to the States. But it was after I left Geigy that I actually started doing something in this area, and the reason for that was that it seemed increasingly possible to me that the studies which he had been carrying out on the role of psychic A&P in the mechanism of action of hormones in breaking down glycogen, and the subsequent studies that Edwin Krebs did showing how psychic A&P brought about the breakdown of glycogen, namely through activating this enzyme which he called cyclic A&P dependent protein kinase. It seemed to me that those studies might have the discoveries made by those studies might have some counterpart in how nerve cells communicate with each other. But it was a good 15 years from the time that I was originally reading that literature till I actually decided to do something about it in the nervous system. Sutherland had shown that the hormones cause the formation of psychic A&P, and that psychic A&P could then mimic the hormones in causing the breakdown of glycogen. This is obviously very important. In fact, he won the Nobel Prize for that work. Krebs then went on to find out how psychic A&P was causing the breakdown of the glycogen, and found that the psychic A&P activated an enzyme which he called cyclic A&P dependent protein kinase, which then phosphorylated and activated another enzyme which was involved in the breakdown of glycogen to glucose. I heard a lecture by Donald Walsh, the first author on that paper. Walsh, Perkins, and Krebs was the paper, talking about the discovery of the psychic A&P dependent protein kinase, and somehow sitting there in the audience, the possibility occurred to me that maybe the work on the formation in response to hormones of psychic A&P and the activation by psychic A&P of psychic A&P dependent protein kinase, which Walsh had just been talking about, might be relevant to how nerve cells signal each other. My concern about this was that hormones can work over a distance of a couple of meters, and the distance between two nerve cells is approximately 1/1,000,000th of a centimeter. So it was not at all clear that this would be the case, but it turned out to be so. The reaction to my initial papers on this subject was not encouraging. It was very widespread skepticism and occasionally outright hostility. There were times that after I give a lecture that people would get up and use adjectives like subversive, which seemed a bit excessive to me. But the interesting thing is that I very rarely had any doubts that this idea was correct. It wasn't, I mean, in retrospect, I think I was probably intellectually a bit arrogant to be so convinced that I was on the right path, but it did help get through the years of adversity. At the time I was a graduate student, there were basically two types of scientists working on the brain. There were biophysicists, electrophysiologists, who thought that one could have a complete accounting of how the brain functions by measuring electrical signals. And then there were, and these people were not interested in the molecular basis of communication between nerve cells. They thought you just explained it all by electricity and molecules be damned. The other group of scientists who did brain research were biochemists, and they would throw a brain into a homogenizer with as much abandon as they would a liver. And they just used the brain as a very rich source of enzymes and lipids and so on. And so these two groups of scientists, the electrophysiologists and the biochemists who chose to use the brain, very rarely spoke to each other. And this is just as well because they didn't have very nice things to say when they did occasionally encounter one another. This debate raged for 40 years with some rather strong personalities involved. It was essentially won by the adherents of the chemical hypothesis, namely that it was chemical neurotransmission, and certainly well over 99% of all synapses are chemical in nature. There are a few that are electrical in nature. So that was sort of the starting point for my own research. I was interested in understanding how neurotransmitters, by activating these receptors, were able to produce a physiological response in the postsynaptic nerve cell. And so we started looking for neurotransmitter-sensitive adenylyl cyclases analogous to the glucagon-sensitive and epinephrine-sensitive adenylyl cyclases, which had been found by Sutherland in his studies of how these hormones break down glycogen. The first such neurotransmitter we found was first such neurotransmitter-sensitive adenylyl cyclase we found was a dopamine-sensitive enzyme. In other words, we took membranes, and in the presence of dopamine, we were able to get the formation of cyclic AMP. And at about the same time, actually about a year earlier, we were able to show that the cyclic AMP-dependent protein kinase, which Krebs had found in the liver, was also present in the brain, but interestingly, it was present in enormously higher concentration in the brain, and of still greater interest, it was concentrated in the synaptic region of nerve cells. So I became more confident then that we were on the right track. I thought that the evidence was highly suggestive, even more than that. A lot of other people didn't think so. And that had its good aspects and its bad aspects. The bad aspects were that, one likes to have one's work appreciated. And it really wasn't, except by very few people. Interestingly, the person who first told me he was convinced I was right was Earl Sutherland. I met Sutherland at a meeting, and he told me that he knew that this hypothesis was extremely controversial. And it was kind of encouraging because he was one of my heroes, and he said that even if I was only 90% correct, it was still a major advance in the field, and so that was a very nice shot in the arm for me to continue my work. Two of the key observations were the demonstration of a dopamine-sensitive adenylyl cyclase and of a cyclic AMP-dependent protein kinase. We then looked for other neurotransmitter-sensitive adenylyl cyclases and found others, most notably a serotonin-sensitive one. And we also found other protein kinases which were not known previously. One was a cyclic GMP-dependent protein kinase, and then a calmodulin-dependent protein kinase. So we now had several neurotransmitter-sensitive adenylyl cyclases, and we had several second messenger-regulated protein kinases. And so the question was, how are these protein kinases working? And so we began to look for substrates for these protein kinases in the brain. And in those days, the techniques for finding phosphorylated proteins were rather primitive. That we had some of the early SDS gels, and we found three proteins, which we call by the imaginative names of protein 1, protein 2, and protein 3. And these turned out to be what we call today synapsin 1. Protein 1 was synapsin 1. Protein 2 was the regulatory subunit of the type 2 cyclic AMP-dependent protein kinase, and protein 3 was synapsin 2. And in retrospect, the reason we found these proteins first is that they're amongst the most abundant phosphoproteins in the brain. And so with the relatively insensitive techniques we had at those times, they were the first peaks to show. After we had discovered in the early 1970s the neurotransmitter sensitive adenylyl cyclases and these second messenger dependent protein kinases, we spent a lot of time trying to prove that they really were involved in the functioning of nerve cells. And because there was so much resistance to these ideas, I spent a lot more time on it than I wanted to. And we used various model systems. For example, we used nucleated erythrocytes from birds, and it showed, for example, that norepinephrine, which regulates ion permeability of the membrane of birds, worked through the psych-AMP dependent pathway. And we used a number of other model systems to prove the validity of the hypothesis. I think that the most convincing experiments came from three studies in collaboration with electrophysiologists. One were studies which we carried out in collaboration with Rodolfo Linas and his students, in which we injected the phosphorylated and dephosphorylated forms of synapsins into squid giant axons and showed that you got these dramatic effects on release. We knew that the synapsins were associated with synaptic vesicles in the presynaptic nerve terminal. And the question was what they were doing there. And what we found in these studies with Rodolfo was that if you injected the dephosphorylated form of the synapsins into the nerve terminal, you inhibited release of neurotransmitter. Basically, you abolished it. It was very dramatic. If you injected the phosphorylated form of the synapsin, nothing happened. If you injected the protein kinase, calcium calmodulin-dependent protein kinase 2, which phosphorylates synapsin 1, then what happened was that you got an actual increase in neurotransmitter release. So our interpretation of this was that the dephospho form of synapsin 1 was inhibitory, and that the kinase, by being injected, converted this dephospho form, which was inhibitory, into the phospho form, which was inactive. And the data supported that idea. Two other studies which we carried out, which I feel helped to influence the neuroscience community as to the validity of our thinking, were studies which we carried out in collaboration with Eric Kandel and Felix Strumwasser. These are two papers which appeared back-to-back in an issue of the Proceedings of the National Academy of Sciences. And in both of these, we were able to show by injecting the catalytic subunit of psychic AMP-dependent protein kinase that we could mimic the actions of neurotransmitters on the target cell. In other words, injection of the kinase catalytic subunit actually could reproduce what happened when you put a neurotransmitter on. So this was rather compelling. We also did some studies in collaboration with Hirsch Gershenfeld in Paris, in which we showed that the psychic GMP-dependent protein kinase was very important, where we could inject psychic GMP or psychic GMP-dependent protein kinase and mimic the effects of the neurotransmitter in those preparations that Gershenfeld had been studying. So gradually during this period, we accumulated evidence with some of the more respected neurophysiologists, and having published these papers with several different groups, people began more and more to accept it. Remarkably, even after this, even after collaborating with some of the leaders in science, I mean, you know, Kandel and Linas and Strumwasser and Gershenfeld were some of the leading electrophysiologists in the country. Even then, there was still kind of a rearguard resistance to the idea that you could actually explain signaling between nerve cells on a biochemical basis. There were two basic reasons for the reluctance to accept some of these ideas. One I already mentioned, that it was that the electrophysiologist types didn't really feel that it was necessary to invoke any biochemical reasoning. In fact, I think Eric Kandel once told me a story about John Eccles being interviewed by the news media and being asked what was the role of biochemistry in understanding brain function, and Eccles replied, absolutely none. And this was not that many years ago. But anyhow, there was a more substantive reason why there was reluctance, and that was that people had trouble with the idea that these biochemical pathways we were studying, which involve enzymatic reactions and have time constants that are of the order of tens or hundreds of milliseconds, could be involved in a process as fast as what was classical transmission, which took, as Lena showed, less than 1,000th of a second to occur. And the answer to this paradox is that the slow synaptic pathways modulate, they modify fast synaptic transmission. We can now think of there being two types of communication between nerve cells, which are called fast synaptic transmission and slow synaptic transmission. The fast transmission takes less than 1,000th of a second. The slow transmission can take tens of milliseconds to tens of seconds. And the best way to understand what's going on in terms of communication between nerve cells is to think of the fast transmission as the hardware of the brain, and the slow transmission involving these protein phosphorylation reactions and second messengers as the software of the brain. I can illustrate this in two ways. Both one example will be presynaptic and one example will be postsynaptic. What we learned from these studies of the synapsons was that the synapsons regulate the efficiency with which a nerve impulse causes neurotransmitter release. In other words, depending on the state of phosphorylation of the synapsons, there's a greater or smaller amount of neurotransmitter released per nerve impulse. So that's a modulation. And that is determined in advance of the nerve impulse by the state of phosphorylation of the synapsons. In other words, presynaptic receptors or calcium entry control the second messenger level and the state of phosphorylation and the state of activity of various kinases, and therefore the state of phosphorylation of the synapsons. So now the system is preset. You've got these nerve terminals with varying amounts of phosphorylated synapsin, and therefore that determines what happens when the nerve impulse comes down to the ending, how many of the vesicles are free to fuse with the membrane and release their contents. In fact, the way it works is that the dephosphorylated form of the synapsons tether the synaptic vesicles to the actin cytoskeleton. When the synapsons get phosphorylated, the tethering is uncoupled, the vesicles are free to fuse with the membrane. And so you can see how something that's very slow, like this phosphorylation and dephosphorylation of synapsons, tethering and untethering of vesicles, is manifested in these very explosive responses, how many vesicles fuse with the membrane when a nerve impulse comes down. The story with regard to the postsynaptic regulation is equally interesting. What happens on the target side of the synapse is that you have these various neurotransmitters which regulate the state of phosphorylation of various proteins in the postsynaptic cell. Amongst these substrate proteins for the protein kinases are the neurotransmitter receptors themselves. And these can be receptors for both fast-acting and slow-acting neurotransmitters. And so what happens is that for example, that virtually all of the biogenic amines and all of the peptide neurotransmitters produce their effects through slow synaptic transmission. Even the fast-acting neurotransmitters, most notably glutamate, which is a fast excitatory neurotransmitter, and GABA, which is a fast inhibitory neurotransmitter, even these fast-acting neurotransmitters produce certain of their effects through these slow signaling pathways. And so you've got virtually every neurotransmitter in the brain is controlling the state of the level of second messengers in the state of phosphorylation of various substrate proteins, one of which, or one family of which, one subgroup of which are the receptors for both fast and slow-acting neurotransmitters. And the way that this affects fast synaptic transmission then is as follows. The phosphorylation of the receptors alters the ability of the neurotransmitter to cause a physiological response in the postsynaptic cell. In other words, when it gets phosphorylated, it either becomes more sensitive or less sensitive to being opened by the neurotransmitter. So we have both modulation at the presynaptic terminal, in which you affect how much neurotransmitter gets released in response to a nerve impulse, and then modulation at the postsynaptic membrane, where the phosphorylation determines how effective the neurotransmitter is in producing the response at the receptor level. In the earlier days in this field, one of the big problems was to determine whether a given phosphorylation had any physiological relevance. It was possible, using high enough levels of protein kinases, to phosphorylate almost any substrate protein. But these could occur, under conditions of artificial conditions, where a very high level of a kinase incubated for a long period of time could phosphorylate practically any substrate protein. And so it was necessary to do two things. One, to show under in vitro conditions, say with purified enzymes, that the amount of the enzyme you're using was relatively low and the incubation time was relatively short. But even more important was to demonstrate in intact cells that the reaction that you were studying was able to occur. This has become increasingly easy to do over the years, because we were able to develop techniques for measuring the state of phosphorylation of proteins in intact cells in a relatively safe way. In the early days, what we had to do is use massive amounts of radioactive inorganic phosphate and get the ATP in the cells labeled, and then try to pick up radioactivity in the proteins that were phosphorylated. But we subsequently developed techniques in which we were able to take phosphorylated proteins or phosphorylated peptides corresponding to those proteins, make antibodies that were specifically that specifically saw the phosphorylated but not the dephosphorylated form of the protein. And then you could do experiments, for example, with intact cells, with primary cell lines, primary cultures or slices, and do your experiment, put a neurotransmitter on, for example, and then extract the tissue and see, using these immunological methods, whether the protein had been phosphorylated and to what extent. And recently, we've even been able to show with intact animals that we can get protein phosphorylation. So the state of the art today is that we can take a drug or a neurotransmitter or a drug and inject it into a whole animal, extract the brain of the animal, and separate the proteins and show that a particular residue on a particular protein was phosphorylated or was dephosphorylated under the particular experimental conditions. So we now have very rigorous ways of testing the validity of the in vitro conclusions. Having found these various second messenger dependent protein kinases, an obvious next step was to try to find the substrates for these kinases. And we found about 100 to 150 bands on gels that were phosphorylated in response to one or another of these protein kinases. There are probably thousands of such substrates. Of this 100, 125, 150 that we found, we've characterized only a handful, because the characterization of a protein is a very labor-intensive thing. After we had found these proteins 1, 2, and 3, which turned out, as I said, to be the synapsins and the regulatory subunit of cyclic AMP-dependent protein kinase, we began to look for substrates which might be present in one type of nerve cell and not in another. And so we dissected out different regions of the brain. I keep saying we, I didn't do any of this. But in this particular case, Angus Nairn and Ivar Wallace dissected out various portions of the brain and then looked for substrates there. The typical way we did it in the early days was to homogenize that piece of brain tissue from that particular part of the brain and add radioactive ATP and then either cyclic AMP or calcium and see whether any proteins got phosphorylated. And that's how we found this 100 to 150 such proteins. Now the next problem came, well, how do you decide which amongst this plethora of proteins you actually study. In the case of these region-specific substrate proteins, we found the largest number of them in the neostratum, the caudate putamen. And again, in retrospect, the reason for that was that the neostratum is a relatively homogeneous part of the brain. The cortex involves many, many, many different types of nerve cells. The neostratum has one prominent nerve cell. It's called a medium-sized spiny neuron. And so a lot of the substrates that we found when we threw cyclic AMP into these homogenates were, as it turned out, localized in this one major type of nerve cell, the medium spiny neuron. Then, say there were about 10 or 12 that were clearly defined on gels. So the next question was, which of these do we follow? And so what we did was to use a trick we'd used when we had studied the synapsins earlier. The synapsins are soluble in weak acids. And so we took this homogenate and looked to see whether any of this dozen substrate proteins might be soluble in weak acid, and one of them was. This is a protein running at a molecular weight of 32,000, and so we called it dopamine and cyclic AMP regulated phosphoprotein of 32 kilodaltons to reflect the fact that it was phosphorylated by cyclic AMP, and as we also showed, it was regulated by dopamine. And it was the dopamine activated the dopamine-sensitive adenyl cyclase that I mentioned earlier, which formed cyclic AMP, which then activated the cyclic AMP-dependent protein kinase and phosphorylated the DARP32. So now we had found these dozen or so substrates for cyclic AMP-dependent protein kinase, and there was every reason to believe that they would also be regulated, like the DARP32 was, by dopamine. So where do we go from here? One of our first questions was which of these dozen molecules to pursue. Since we'd been able to get a pretty high degree of purification of DARP32 by a simple expedient of acid extraction, it seemed worthwhile pursuing this one rather than some of the less purified proteins. So what do we do with the DARP32? Well, you want to know, for example, what happens when you phosphorylate it. Where is it phosphorylated, and how does this affect its properties? So we identified the amino acid residue which was phosphorylated, and we were able to determine the biological effect of the phosphorylation by the fact that DARP32 was analogous in its sequence to another protein called protein phosphatase inhibitor 1, which had been found, I think it was in muscle, and had been shown in its phosphorylated form to be an inhibitor of protein phosphatase 1. So we undertook an investigation to see whether DARP32 might indeed, in its phosphorylated form, inhibit protein phosphatase 1, and that turned out to be the case. The phosphorylated form of DARP32 inhibited protein phosphatase 1, whereas the dephosphorylated form was completely inactive. So now the question was, can you show the same thing in brain slices, in intact cells, whole animals, and we were able to do all that. Show that dopamine or cyclic AMP analogs like dibutyl-cyclic AMP were able to phosphorylate the DARP32, and this was then able to inhibit protein phosphatase 1. So we now wanted to know whether other molecules, other neurotransmitters might regulate DARP32. And the reason for that was that dopamine and glutamate both have very profound actions on these dopamine receptive neurons. Dopamine in most circumstances is inhibitory, glutamate is always excitatory, so we wanted to know whether glutamate, like dopamine, might affect the state of phosphorylation of DARP32. And we found indeed that glutamate did affect the state of phosphorylation, but interestingly, it did it in opposite direction to the dopamine. The dopamine increased phosphorylation and therefore increased protein phosphatase 1 inhibition, whereas the glutamate decreased phosphorylation and therefore decreased protein phosphatase 1 inhibition. That is to say, it made it the protein phosphatase 1 more active. We then wanted to see what the physiological significance of the DARP32 protein phosphatase 1 cascade was. And we approached this in two different ways. We injected into nerve cells various protein kinases or protein phosphatases or activators or inhibitors of these enzymes, and were able to show that these compounds that we injected into the nerve cells either mimicked or antagonized the various neurotransmitters consistent with the schemes that we were developing. The other approach we used was to knock out the DARP32 gene. This is work of Alan Feinberg. And he then characterized this in collaboration with a dozen leading neuroscience laboratories, various kinds of electrophysiologists and so on, and behavioral people. And we were able to show that all of the effects of dopamine were abolished in these DARP32 knockout mice. That means electrophysiological effects or behavioral effects or biochemical effects. So it was like a dream phenotype. You always worry when you do these knockouts that they'll either be, the knockout will be lethal or it'll have no phenotype. But here we had a viable mouse with a beautiful phenotype, all sorts of interesting changes. Not only were all the effects of dopamine abolished, but also all the effects of the antipsychotic drugs were abolished, and the effects of various drugs of abuse, such as LSD, opiates, marijuana, heroin, etc. In addition, all these drugs of abuse regulated the phosphorylation of DARP32, so that their ability to regulate DARP32 phosphorylation, together with the fact that in the DARP32 knockout mice these drugs of abuse no longer work, clearly indicated that DARP32 is a central molecule for the drugs of abuse acting. Once we knew that dopamine and glutamate had these opposing actions on DARP32 phosphorylation, which was very exciting because there were also opposing actions of dopamine and glutamate on the electrophysiology of these cells. We then undertook a study of all of the other neurotransmitters known to affect the physiology of these cells. And what we found was that every neurotransmitter which affects the biology of these cells regulated the state of phosphorylation of DARP32. But this, it turned out, was just the tip of the iceberg, because there were several other regions of the DARP32 molecule which contained consensus sequences for phosphorylation by various protein kinases. And so we wanted to see whether these other kinases would indeed phosphorylate the DARP32 on these other residues, and indeed they did. And we know today of a total of 4 phosphorylation sites that are regulated by various kinases. So then, to summarize what I said so far about these modulatory sites, we have casein kinase 1 phosphorylating serine 137, which now prevents calcineurin, PP2B, from dephosphorylating threonine 34. We have casein kinase 2 phosphorylating serine 102, which potentiates the ability of pKa to phosphorylate DARP32. So they are both potentiating the effects of dopamine. In the one case by inhibiting the dephosphorylation, in the other case by potentiating the phosphorylation. And these are both substrate-directed effects. They affect the structure of the DARP molecule so that it becomes a poor or a better substrate for a kinase and a phosphatase. The third modulatory pathway, to me in many ways, is the most interesting, and that is the phosphorylation by CDK5 of threonine 75. When you phosphorylate threonine 75, you turn the DARP32 molecule into an inhibitor of pKa. In contrast to the other 2 phosphorylations, which change the structure of the substrate so that it's a better or poorer substrate, the threonine 75 is actually an inhibitor of pKa. So the pKa not only can't phosphorylate DARP32, it can't phosphorylate any other substrate. This means, among other things, that the DARP32 is a bifunctional molecule. Depending on whether it's threonine 34 that's phosphorylated or threonine 75 that's phosphorylated, DARP32 is either a phosphatase inhibitor or a kinase inhibitor. And you will appreciate that this has profound effects on the biology of the cells, if you're shutting off the kinases or shutting off the phosphatases. And you're doing so globally because both pKa and pp2b have a wide spectrum of substrate proteins. The system wouldn't have to have been rigged that way. It could have been constructed in other ways. So you could have had something that controlled all the kinases and something else that controlled all the phosphatases in one molecule, which is kind of nice because you turn this one on and shut this one off. All right, let me think about this. So really what happens, it's not so simple like you're turning on all the kinases or you're turning on all the phosphatases or turning off all the kinases. What you're doing is a battle between the CDK5 PP1 pathway. Activation of CDK5 activates PP1. Activation of pKa activates PP2A. So you can't say it's the kinases against the phosphatase. It's a pKa PP2A pathway against the CDK5 PP1 pathway, and it really gets. I'm still thinking through the ramifications of this. Yeah, we found the DARP32 in early 80s, I think like 1982 or something. We published the first paper. And at that time, they determined the phosphorylation sites of proteins. You had to sequence the molecule. And to determine the structure of proteins, you had to sequence the whole molecule by amino acid sequencing, which is extremely laborious. And nowadays you just, clone them and sequence the DNA, so it's much easier. So as we began to build up this picture that not only dopamine and glutamate regulated the state of phosphorylation of DARP32, but found that all these other neurotransmitters which regulated the physiology of these cells also regulated the state of phosphorylation of DARP32. So that was one body of evidence we had. Then we had found these other 3 phosphorylation sites, so we now knew that there were 4 phosphorylation sites. And we began to study the neurotransmitters which would regulate those. And so you start putting together a pretty complex picture of intracellular signaling. And the more information that we obtained, the more interesting new questions there were. For example, the schemes we developed indicated that the receptors for fast excitatory transmission, namely for glutamate, the glutamate receptors, the AMPA and NMDA receptors, regulated the DARP32 PP1 cascade, and that the DARP32 PP1 cascade regulated the state of phosphorylation and activity of these glutamate receptors. So we wondered whether the PP1 might be localized in the region of the brain, in the region of the nerve cell where the receptors are located. Where are the receptors located? They're located, the vast majority of excitatory glutamate receptors are located in the spines on the dendrites. And so we made recombinant PP1 and made antibodies to this recombinant PP1 and then localized the PP1 and found, lo and behold, that the PP1 was also concentrated in the spines. Then one of my colleagues, Patrick Allen, using Yeast-2 hybrid technology, looked for a chaperone that might be responsible for targeting PP1 from the cell body to the spines. He found such a molecule, which we called spinophyllin. And then we made antibodies to the spinophyllin and then localized the spinophyllin. And it turns out to be in the spines, not only in the spines, but in the spine heads, not even in the spine necks. This is precisely where the fast excitatory glutamate receptors are located. So it was in the right place, and this provided more evidence. Now, the spinophyllin itself is a substrate for regulation by phosphorylation, and it's certainly regulated by pKa in all probability, although we haven't proven it yet. It's regulated by dopamine. So here's another regulatory switch. The spinophyllin determines how effective the dopamine is in controlling the glutamate receptors. So as the work has evolved, it turns out that there are more and more areas of crosstalk between these different neurotransmitters through these protein phosphorylation signals. We have all the neurotransmitters regulating 3 and 34. We have many neurotransmitters regulating these other phosphorylation sites, interacting in very complex ways. For example, glutamate, by activating CDK5, controls the dopamine pathway, and dopamine by pKa, activating pKa, controls the glutamate pathway. And these systems have many built-in mutually antagonistic components. So we now know maybe 25 different interactions just in the dopamine, glutamate, and closely related neurotransmitter complex. This is, since many therapeutic drugs work by controlling the efficiency of neurotransmitter release. In fact, almost all neuroactive and psychoactive drugs, there are some exceptions like general anesthetics, local anesthetics, but most of the drugs we have in the clinic today, antidepressants, antipsychotics, antiparkinsons, and so on, work at the level of neurotransmitter receptors. And in fact, what's been going on largely in pharmaceutical industries is to look for agonists or antagonists of the actions of neurotransmitters on their receptors. One of the reasons I'm pleased about the work we've done is that it's opened up a whole new world of potential targets. If you have a neurotransmitter activating receptor and you find 14 different biochemical components that are involved in mediating the actions of that neurotransmitter activation of that receptor, it provides you with 14 new targets for potential new drug therapy. And I think that over the next number of years that the drug companies are going to be more and more trying to find medicinal agents which target these intracellular targets. The argument can be raised that the intracellular machinery is so complex that wouldn't it be foolhardy to go in and try to perturb it? But in fact, what you're doing when you put an agonist or an antagonist for a neurotransmitter on the outside of the cell is to perturb all that intracellular machinery anyhow. You just don't know what you're doing. It's like an ostrich with its head buried in the sand to say, oh, this is simple to be outside the cell here at the receptor level. We don't want to know what happens once the receptor is activated. But the same system that's being perturbed by regulating the receptor is being perturbed when you go intracellularly. There's already there are already a number of examples of the benefits of intracellular targeting. One example of how intracellular targeting could be more effective is the following. It comes from the Parkinson's disease literature. Levodopa, which is the first-line drug of choice in the treatment of Parkinson's disease, is very effective in most patients with Parkinson's disease initially, but after a short time, these patients many of them become refractory. One hypothesis as to why patients become refractory to levodopa is that the levodopa causes a downregulation, a desensitization of the dopamine receptors. How attractive then it is to think of going intracellularly, bypassing the receptor and potentiating the dopamine signaling pathway. In theory, at least, we should in this way be able to develop drugs which do not have this loss of sensitivity, this tachyphylaxis effect. And therefore, hopefully, we will have drugs that are as effective as levodopa without the loss of effectiveness. About 10 years ago, a scientist by the name of George Glenner took plaques from the brains of patients who had died from Alzheimer's disease and studied the composition and found there was one protein there which he called beta amyloid. And he published that sequence. And then shortly thereafter, several laboratories using that sequence cloned what was then called the amyloid precursor protein, which was shown to be cleaved into formation of beta amyloid. So the amyloid precursor protein, as the name implies, was a protein, the breakdown of which gave beta amyloid. When that sequence was published, my colleagues and I thought this would be an interesting area to look into. Why? Because in the amyloid precursor protein sequence, there were consensus sequences for phosphorylation by various protein kinases. So we began to study the possible regulation of beta amyloid formation by regulation of protein phosphorylation. What we found was indeed that if you put on protein kinase activators such as phorbol esters, or protein phosphatase inhibitors such as ocadaic acid or colliculin A, that you could greatly reduce the formation of beta amyloid. If you combine a protein kinase activator and a protein phosphatase inhibitor, you can reduce beta amyloid formation by over 90%. So this is very exciting. The problem is that these kinase activators and phosphatase inhibitors are tumor promoters. So obviously that's not the way to go. And there are good ways to explain why they're tumor promoters, because these protein phosphorylation pathways are involved in cell replication. We then have spent a fair bit of time elucidating how protein phosphorylation controls the synthesis of beta amyloid. One way in which it does it is by controlling the transit time through the ER and the Golgi of the amyloid precursor protein. The amyloid precursor protein is converted into beta amyloid in part in the Golgi, and what these these agents which increase protein phosphorylation do is increase the speed of transit through the Golgi. So they have the APP, the amyloid precursor protein, has less time to be exposed to the enzymes which form the beta amyloid. That's one way in which the beta amyloid is reduced. We've also been able to show that various hormones, which have been, the lack of which have been implicated clinically in Alzheimer's disease, are also involved in regulating beta amyloid formation. For example, women, postmenopausal women given estrogen treatment have a greatly reduced incidence of Alzheimer's disease. We were able to show that treating cells with estrogen reduced beta amyloid formation. And in fact, we could take cytosol from cells that have been treated with estrogen and add it to naive cell extracts and show that that cytosol contained a factor in it produced by one or more factors produced by the estrogen, which enabled the reduction, which brought about the reduction of beta amyloid formation. So there are one or more factors there which mediate the actions of estrogen in lowering beta amyloid formation and almost certainly in reducing the incidence of Alzheimer's disease in postmenopausal women. We've had three major areas of interest over the last 20 years. When we first started studying protein phosphorylation in the nervous system, there was like this whole world in front of us. And we were the only laboratory in the world working on protein phosphorylation in the nervous system. So we could just go around at a leisurely pace and study this or this or this aspect. As more laboratories entered into this research area, we focused down, and we focused down in three areas. One is the dopamine signaling and the related story in the basal ganglia. A second is the Alzheimer's, and the third is the role of synapsins in regulation of neurotransmitter release and in new synapse formation. Increasingly, we've been focusing on the dopamine basal ganglion story and the Alzheimer's story, and just because it's too much, and so probably eventually I'll just narrow down to either the dopamine signaling or the Alzheimer's story, because as each of these stories is developed, there's more and more things to do within it, and I like to go deeply into things if I can, not be too broad, but rather deep. The priorities of these three subjects were not always this way. For quite a while, the synapsins were what intrigued me the most, but you know, one moves in one's research programs into those areas which look the most exciting. What I try to do is work on the most exciting doable project. And there's some projects that are extremely exciting, but you feel you can't really solve them. So I try to take those projects which are most exciting and that I think can be solved and work on those. And it's just that it's not a reflection about synapsins. There's so many pathways have been uncovered in this basal ganglion, the neostratum story, that are crying to be studied. And the other reason is that the synapsins were the first synaptic vesicle proteins found, but there's a number of very excellent younger scientists today, some of whom studied in our laboratory, who are working in that area, and I've never wanted to work in competitive areas. And the synaptic vesicle protein area has some extremely fine scientists in it, but it's also a highly competitive field, and so that's less attractive to me. Well, I've been very fortunate over the years of having a really wonderful and wonderfully talented group of young colleagues associated with me. It's interesting, it's a case of the rich getting richer. If you publish a few good papers, you start attracting excellent younger people, and the more excellent younger people you have, the better your work becomes. The better your work becomes, more excellent people you get. So it's sometimes I think that the laboratory is better off without me around. There's so many brilliant young people, and have been over the last 25 years, that who have contributed so much. Most people are here for a period of 3 or 4 years, 2 to 5 years. One colleague, Angus Nairn, and I have worked together for over 20 years, and I think our skills and personalities complement each other very well. He's in the process of moving now to Yale University, where he's going to be a professor in the departments of pharmacology and psychiatry. Angus and I have worked, extremely well together, and he's helped me running the laboratory, and we have we have about 25 to 30 people in the group. I don't particularly like a larger group. In fact, I prefer a smaller group, but our work is so interdisciplinary that you need people who are molecular biologists or protein chemists or enzymologists or cell biologists or neuroanatomists. You need all these disciplines to work in a concerted fashion on a project. And so we have more people than I ideally would like to have. But we work together pretty well as a team, and a lot of big laboratories try to put their younger people in competition with each other. I mean, the one very famous Nobel Prize-winning enzymologist who would say, you will purify this enzyme from pig, and you will purify this enzyme from sheep, and then they put them into a cockfight, and these people would, keep their data secret from each other. Horrible. And I take a very dim view of people behaving that way. I get quite annoyed if somebody is not cooperative in the laboratory. And by and large, it works very well because people help other people in the laboratory, but they get helped a lot too. And I actually prefer to have, it's essential from my point of view to have people who are get on well, who like to be interactive and talk creatively together. I think that it becomes a very synergistic situation. And I have kept up with many of the people that have gone through the laboratory. Many of them we still collaborate. I have colleagues in Japan, Italy, Sweden, France, Germany, China that we still are working pretty closely with, which is very nice because it's like one large family. And when the people in the laboratory travel to other countries, there's always somebody that they can stay with who can help them with their travels and so on. And it's very nice. It's a very familial kind of feeling. And people come back to visit the lab a lot, and I love that. And it's just been great.